Welcome to Tell Me More Stories Told Us podcast.
Great to be here.
Today, we're diving into something you've probably seen, maybe even used, without thinking much about it. The eyedropper.
That's right. Or, the Pasteur Pipette, as it's more formally known. Or
That's
just a dropper.
Exactly. Seems really simple, doesn't it? Just a little tube and a bulb.
It does seem simple, but like a lot of things, there's actually quite a story there. Its history, the different types, how it's used. It's surprisingly versatile.
So that's our mission for this deep dive then. Unpack this unassuming little tool.
Pretty much. We'll explore where it came from, what it looks like today, and some of the, well, frankly, quite surprising things it can do. You might have a few aha moments.
I'm ready. So let's start with the basics. What is it, fundamentally?
At its heart, it's really just a tool for moving small amounts of liquid from one place to another.
The classic picture in my head is that thin glass tube, tapered down to a point.
Yeah, that's the pipette part. And then the rubber bulb on top that you squeeze. That bulb is actually why some people call it a Teat pipette.
Oh, okay, I can see that.
Huh.
A bit descriptive.
Hey, it is.
Yeah.
But yeah, that's the classic look. But it's not the only look.
Right, you mentioned different types.
Yeah, you get them in plastic, too. Loads of different styles in both glass and plastic nowadays.
Okay, let's rewind a bit then. History. You said Pasteur Pipettes. That's the Louis Pasteur.
The very same. The famous French scientist. He apparently used a version of these quite a lot in his research back in the 19th century.
Wow. So this isn't some modern invention.
Not at all. And think about labs back then. They didn't have all the fancy specialized gear we have now.
Right, they'd have to improvise more.
Exactly. And a key thing for Pasteur and others at the time was transferring chemical solutions without, getting stuff from the air mixed in.
Ah, contamination control. Even back then.
Absolutely crucial.
Yeah.
Especially for experiments where purity was everything. You needed reliable results.
So the dropper helped with that.
It did. Because you could sterilize the glass ones, pop a rubber bulb on,
Right.
and move liquids around without exposing them too much. It was a basic but really important way to maintain some level of sterility.
That really highlights how important it was right from the start. Okay, so let's fast forward. Glass versus plastic. What are the main differences there?
Okay, so glass Pasteur pipettes, they're usually made from one of two main types of glass, borosilicate or soda lime.
Borosilicate. That sounds familiar from lab glassware.
It is. It's tough stuff. Stands up well to heat and most chemicals you find in the lab. And it's generally pretty economical because it's easier to work with when making the pipettes.
Okay, and soda lime.
Soda lime glass isn't quite as resistant, chemically speaking. But for something inexpensive like a Pasteur pipette, it does the job just fine.
Gotcha.
And here's a fun little detail, a bit of lab craft, really. You can actually take a glass pipette and carefully heat the tip in a flame to draw it out, make it super fine.
Oh, really? Why would you
Yeah.
do that?
Scientists use these modified pipettes as spotters for a technique called thin layer chromatography. It lets them apply tiny, precise spots of a sample. Pretty neat bit of adaptation.
That is clever. Okay, so that's glass. What's the deal with the plastic ones? Transfer pipettes, you called them.
Right, plastic Pasteur pipettes or transfer pipettes. These are usually just one single piece of soft plastic, often polyethylene.
So the bulb isn't separate.
Nope, it's all molded together. The bulb part is usually thinner, easy to squeeze, and the stem part, the tube, is a bit more rigid.
And sizes.
They come in common sizes, maybe holding 1 milliliter up to 5 milliliters, something like that. And they're designed to give a sort of approximate drop size, say between 10 and 50 microliters per drop.
Approximate, though. Not super precise.
Definitely not for high precision. You'll often see some rough volume markings printed on the stem, like 0.5 milliliter, 1 milliliter, but they're really just guidelines, not for accurate measurement.
So what's the big advantage of plastic then?
Well, the main things are cost and disposability. They're cheap. So cheap you just use them once and throw them away.
Ah, so that avoids washing or potential cross-contamination between samples.
Exactly. That's huge, especially in biology or medical labs where you're handling lots of different samples. Also, sometimes delicate things like cells or proteins can stick to glass. Plastic can sometimes reduce that loss.
Interesting point.
And some plastic ones have really long, thin, flexible tips, great for getting into tiny little tubes or vials.
Any downsides to plastic?
Well, they're mainly for water-based solutions, aqueous media, common in biology.
Yeah.
Strong organic solvents can actually dissolve or damage the plastic.
Ah, okay. Good to know.
And yeah, as we said, they're just not built for the kind of precision you might need sometimes. compared to more calibrated glass pipettes.
So it really depends on what you're doing. Okay, let's say I need to use one, maybe just adding some reagent drops. How do I do it properly?
Right, so first off, remember, it's not for measuring exact volumes. It's for adding drops, roughly controlled.
Got
Always give the tip a quick look first. Make sure there aren't any cracks or chips that can mess up the drop formation.
Safety first and accuracy.
Yeah. Now, if you want to be a bit more consistent, not super precise, but more consistent, a good tip is to rinse the pipette once with the liquid you're about to use.
Okay, like priming it?
Kind of. Just draw a little up and squirt it out into waste. Then, to draw up your actual liquid,
Right.
squeeze the bulb before you put the tip in the liquid. Push the air out.
Right.
Then, put the tip into the liquid, keep it vertical, and slowly, gently release the bulb. The liquid will get drawn up.
Slowly is key.
Yeah, and try not to overshoot. Don't suck the liquid way up into the bulb itself. That's a common way to contaminate the bulb or your sample later.
Keep the liquid in the stem part.
Okay.
And dispensing.
When you're ready to let the drops out, touch the tip against the inside wall of the container you're adding to. Hold it at maybe a 30-45 degree angle. That helps the drop release cleanly.
Against the side, not just dropping into the middle.
Generally, yeah, it gives better control. And super important safety point. If a glass one breaks, don't just toss it in the regular trash. It needs to go in a special container for broken glass or sharps.
Definitely noted. Okay, so that's the basic function covered. But you hinted at more surprising uses earlier. This is the tell me more part.
This is where it gets really inventive. These simple pipettes get adapted for all sorts of microscale work working with tiny amounts.
Like what?
Okay, one example. Microscale column chromatography. You can take the narrow end of the pipette, just above the very tip, and carefully stuff a tiny plug of cotton wool or even tissue paper in there.
Okay, why?
Now you've basically made a tiny filter column. You could put your sample in the top, maybe add some solvent, and filter small amounts of liquid. You could even use the bulb to gently push slightly thicker solutions through.
Wow, a DIY mini filter. That's really resourceful.
Isn't it? And chemists can take that even further. With some practice and the right packing material like fine silica gel, they can actually do something called microscale flash column chromatography. It's a purification technique, all inside a little Pasteur pipette.
Incredible. What else?
How about microscale distillation?
Distilling with an eyedropper? How?
Okay, imagine you have a tiny amount of liquid you need to purify by distillation. You heat it gently in a small tube. The vapor rises. You take a cold Pasteur pipette, squeeze the air out, and insert the tip into the warm vapor zone.
Ah, so the cold tip acts as a condenser.
Precisely. The vapor hits the cold glass, turns back into liquid, and collects right there inside the pipette tip. You've just done distillation on a miniature scale.
That is seriously clever, like a tiny chemistry set.
It really shows the ingenuity, doesn't it? And another one, simpler maybe, storing tiny liquid samples with a plastic pipette. Once you've drawn up your sample, you can use heat carefully with a heat sealer or even a flame sometimes to just melt the very tip closed. Seals it right up. Now you've got a tiny sealed vial holding your sample.
Perfect for preserving a small amount of something.
Exactly. And maybe one of those critical but less visible uses is in medical labs.
Oh, yeah. How so?
Well, we said they deliver a relatively consistent drop volume, right? Even if not perfectly precise. That consistency is actually really important for certain types of tests. Think about drug screening tests or observing blood cell characteristics, things where adding a consistent small drop is key to getting a reliable result.
So even though they aren't precision instruments, that good enough consistency is vital in diagnostics.
Absolutely. In fields where small differences matter, that reliable drop can be crucial.
So let's recap. Basic liquid transfer, sure, but also mini filters, mini stills, trainee storage containers, and a key tool in medical diagnostics. That little eyedropper really punches above its weight.
It really, really does. It's amazing how this simple design, born out of necessity maybe centuries ago with Pasteur,
Yeah.
how it just keeps finding uses, adapting, and remaining essential in labs everywhere.
It truly makes you appreciate the cleverness behind seemingly simple tools.
It really does.
Yeah.
And, speaking of appreciating these deep dives, if you are enjoying exploring the stories behind everyday things like this,
Now's a good time to mention it.
yeah, definitely hit that subscribe button. That way you won't miss any future explorations we do here on Tell Me More.
Absolutely. We love uncovering these hidden histories. You never know what we'll look into next.
So true. This whole discussion makes me wonder,
What's that?
It just raises the question, doesn't it? What other things around us, things we think are totally mundane, might have equally fascinating stories, equally surprising hidden depths?
Hmm. That's a really interesting thought. Like, what's the secret history of the paperclip? Or something else equally common?
Exactly. Maybe the next time you pick up an eyedropper or something similarly simple, you'll just pause for a second.
And see it a bit differently.
Yeah, see it with new eyes, appreciate its journey, maybe its quiet contributions to science or medicine or just everyday life. There could be a whole universe of detail waiting right there.
A fantastic thought to end on. It reminds us that curiosity can find amazing stories anywhere. Thanks for joining us on this surprisingly adventurous look at the humble eyedropper.
My pleasure. It was fun.
Until our next deep dive, keep looking closer.